done in different laboratories. There's extensive postdoctoral work. Right now, he's in the Birla Institute of Scientific Research, Jaipur, and he's focused research on known and unknown regions of human genome. So, Dr. Suravajhala. So, we will restrict 20 minutes. I would be, if you would like to have some discussion, and therefore, you would like to restrict your time. Thank you.
Thank you so much, David Chase, and thank you, Jan, for this wonderful invitation. So I'd like to give a talk on this prostate cancer evolution that my lab has been doing over the last 6 years. I come from Amrita Vishwavidyapitham, which is somewhere in the southwest coast of this Malabar region, between Trivandrum and Kochi, a beautiful campus, lush green campus, between these wonderful, what you call backwaters. Our chancellor is Mata Amritanandamayi. She is the head of civil society's C20 chairperson. And our Amrita School of Biotechnology is with 40 plus academic staff, and Dean is Dr. Vipin Nair. All faculty are coming from US or Europe, and ours is rated the best private university in India. So this is an ode to Rajit Singh. 21 years ago, when I joined CCMB as a young project fellow, I was sitting in the audience, and as a curious person, I asked some questions. He told me one thing before he invited me to his group as a small project fellow. Prash, you need to keep asking questions, and then you should be an admitted speaker. And today, after 21 years, I think, thanks to Gyan, for this wonderful opportunity that is rendered. And we have always, asked about ants, what ants do. We step on them, we kill them, knowingly or unknowingly, and yet, they come together. And the leader of those particular ants always, guides the path, and that's where, Laji is. And this is an absolute fulfilling. So today, I'm just going to give a talk on some aspects where I'll split my talk into 3 small parts, where we have been doing largely on the role of, CA prostate and regulatory genomics. And if we really talk about any organisms, there are always contexts. If we really talk about plants are suicide, they invite a lot of stress. We as humans keep moving, we take up stress, we come back to stress, we identify certain raise and detrain behind or overcoming these particular stress and all those kind of things. But at the end of the day, what matters is whether or not we are trying to ask certain relevant questions in taking up the true positives, false positives, true negatives, and false negatives. So if we really talk about central dogma of bioinformatics, sequence predicts structure, structure predicts function is in the realm. But at any given point of time, at the epithet of this, you find a lot of intrinsically disordered proteins where the sequence need not delve out necessarily a kind of a structure. It could, in principle, be unstructured. That's where we have unfunction, and that's where we have all those kind of diseases. And that's where systems biology has really grown up over the years, taking cognizance of bioinformatics, where it has really more of the value chain. Thanks to wonderful entities like the mutations arising and falling, there have been a lot of proteins evoluting in the value chain and all those kind of things. So the first part of my particular talk is largely into prostate cancer mutations relevant to India. This is what I delved upon after I became independent in 2016 when I joined Birla Institute of Scientific Research. So this comes with an adage, with a prelude, that when we eat normally, we tend to be very healthy. And we eat less, some pale people become very unhealthy. But when we eat again in optimally different time intervals, we are again very healthy. But in principle, when we eat too much, we not only become unhealthy, we might also become obese. But that's not the stop goal. We also tend to become prostate cancer victims when we turn out, after 50 years. So this is where, the rationale was set in when I was doing my 8th year of my postdoc in Denmark, where my colleague, was overfeeding these particular cows and bulls so as to get, better semen and better, milk. And the poor bulls, they moan in pain just because they're not just obese, their androgen receptor pathways are going, in a different time zones. And rationally, it is also said because when I moved back, we were working on something called Asian Indian type 2 diabetes mellitus. We dealt upon a wonderful phenome interactome network in Asian Indian type 2 diabetes, where we identified those differentially expressed genes for both, for three of these particular phenotypes, so type 1 obesity and type 2 diabetes. And this was where we found one long intergenic non-coding RNA as a candidate marker regulating this type 2 diabetes. So moving on, then as a part of the third part, I'll again come back to this particular slide, and then I founded this CAPC, See a Prostate Consortium of India, where we pulled in about 31 scientists all over India and also across different countries and started performing this prostate cancer whole exome sequencing analysis. So if you really look into the prelude for this, many African Americans eat red meat. They eat hard-boiled meat. That's why that's probably one of the reasons why prostate cancer is caused in them. As opposed to Asian Indians, about 63% of Asian Indians are largely vegetarians, and the incidence is really growing. And I'll probably skip here. We have benchmarked our own whole exome sequencing pipeline in our lab to simply give you FASTQ rates, and after the pipeline is set in, it will give you the list of mutations prioritized for that particular deceased phenotype. And this is a Mina et al. 2018. So what we observe in this particular instance is, as you can see here, the square boxed genes are those particular genes that are relevant to Caucasian, and the circle ones are all relevant to Indians, the others being piggybacking between other countries as well. As you would see here, BRCA1 and BRCA2 is well known. BRCA2 is also known in Caucasian population. But apart from that, from our Indian perspective, we found mutations that are largely associated with OPA, largely associated with mitochondrial biogenesis, largely associated with TP53, and largely associated with myo15A, which is related to gut microbiota. So this begs us a question that what we swallow is what we get. It's something like a Wiswick principle, which means what you eat is what is that you're trying to get. So there's something to do with gut microbiota that is emanating this prostate cancer biogenesis. So the first part of my particular talk is, incidentally, from a small cohort of 25 population we did in 2018. Now it has been extended to about 90 cases. All patients, most of them, 95% of the patients are vegetarians, and we identified certain diffident mutations largely responsible for mitochondrial biogenesis, gut microbiota, the vitamin D receptors, and all those kind of things. And we developed a kind of a conceptual framework, for prioritizing these particular mutations, specifically to prostate cancer phenotype in India. So the second part of my story is, we thought, we asked another question: if these prostate cancer causing genes are there, are these regulatory in nature? Which means, if we find a gene that is causal to the PCA phenotype, would that particular gene be, interacting with any of its, a gene, sitting next to that? So for this, we employed something what we call it as a genome walking study, which means, let's imagine we have a mutation somewhere sitting here. Earlier, we have prioritized certain SNPs in such a way that if a SNP is sitting somewhere here, somewhere 5' UTR of this particular region, we find a lot of, transcription factor binding sites, promoters, open chromatin elements, CTCF motifs, and further upstream we have something called non-coding RNAs, where genetic variation is largely sitting in there. So we walk through each of those particular mutations that are specifically causal to prostate cancer, and we try to identify what are those particular mutations that are also sitting in long non-coding RNA influencing the PCA phenotype. This is also supplemented by RNA-seq analysis, keeping in brevity of time and not talking about that particular aspect. And that way, we found not only a long non-coding RNA, but also pbRNAs, which are a class of another small non-coding RNAs, which are anywhere between 35 to 40 bases in length. And what we have just recently communicated was, we found these particular differential expressed genes, including the long non-coding RNA. One of those well-known long non-coding RNA is sitting somewhere here. But through this cytohubba network, what we came up with an analysis is, the brighter nodes, which means larger the color node, is greater is the chance that that particular gene is largely differentially expressed. So that's where you can see all those maroon nodes. Those maroon nodes are largely differentially expressed, which means they're largely upregulated most of the time, that are interacting with those other genes. So we came up with this coefficient network and came up with certain degrees centrality and closest centrality and validated them using the functional genomics approach. So the second part of this particular talk is we found not only long non-coding RNAs that might be regulating with respect to PCA biogenesis and Indian phenotype. We also found certain pDRNAs that are largely associated with these particular conditions. And a couple of these long non-coding RNA protein interactions, we are performing a CLIP-seq assay, which is a cross-linking immunoprecipitation sequencing to check whether or not they're physically interacting, which we are currently doing this post-COVID, so that we can consider them as plausible biomarkers. So the third epithet of my particular talk is what does systems bioinformatics can entail, taking these two particular stories that we have. So my PhD was largely on hypothetical proteins when I started in 2004 in Denmark. I always worked on something what we call it as the known unknown regions, which means you know that a protein exists, but you don't know what the function it does. So we were always looking into the syntenic regions, whether or not a gene of interest is there, whether or not it is syntenic with certain region of interest that is probably well annotated. Say, let's take the example of cows, or maybe chimps, or orangutans, or whatever. So those kind of things we really asked, and we developed a kind of a semantic called as hypothome. Hypothome is an interactome of hypothetical proteins, which means if a known protein is interacting with an unknown protein, the unknown protein would take up the function of that particular known protein, and that is where we have taken that. So keeping in view of this particular cognizance, now that we have done about 90 exomes and about 8 RNA-seq whole transcriptome short-term sequencing approaches, we are just trying to contemplate whether or not is there any kind of an introgression that could be seen with respect to this PCA genotypes. That's because prostate cancer is largely positive to those particular people who eat non-vegetarian diet. And Denisovans, we have these Neanderthalenses, they're largely meat-eating people. They survive in those particular different conditions. So are there any predisposable genes that have been introgressed over the years, over the last probably at least 15K or 20K years ago, which we just started working on it. And just a couple of days ago, there is Illiton. This paper is just in the press. It just came last week. They have wonderfully developed something called a comparison of non-introgressed haplotype in lieu of, say, liver cancer, as well as to those introgressed or well as non-introgressed haplotypes with respect to the liver cancer mutations. Which means there could be a chance that our theory, the kind of work that we are trying to do, taking up the prostate cancer, might have chance taking up these kind of questions, which I would like to probably collaborate with one of you on these particular directions. So we wanted to ask, is there a role of Neanderthal introgression in prostate cancer? And that way, this Neanderthal introgression will provide an opportunity to identify risk attributing mutations in lieu of diet and as well as lifestyle. So that thing we pretty well know. So that's where we could probably tend to ask a relevant question whether or not any of these particular mutations tend to be purifying in nature, as opposed to any of those particular non-synonymous substitutions which might develop completely a pan lineage of introgressed mutations sitting somewhere here. So the final part of my particular talk. So we have done quite a number of whole exome sequencing and PCA with respect to Indian phenotype from our consortium. We have part 2 where we have done the regulome-based studies, the regulatome-based studies, where we have done some RNA-seq as well, because getting the RNA from FFP blocks is quite a cumbersome approach. So we could do that. I didn't want my PhD fellows to run away, so we kept some of those particular samples for fresh protein samples. The part 3 by 2024, we tend to come up with this deep variant-based approach, where we want to come up with a machine learning heuristics. Maybe after 300 exomes, by which time will be done, we wanted to come up with a kind of a hypothesis, say from 301st exome, can we say that these particular mutations could be seen or probably introgressed by lineage. So this is what we wanted to come up, and as you can see, a lot of prostate cancer is largely burgeoning. A lot of money is need to be spent for treatment and all those kind of things. And that's where MALAT1 is a well-known long non-coding RNA, which was known as a genetic biomarker. Like this, we've already identified a couple of long non-coding RNAs, but we tend to expect some long non-coding RNAs to be coming into this. I'm done, sir. And then our mission is to come up with no more biopsies by 2028. This is our milestone that we wanted to meet, so that we can come up with a simplest lateral flow assay at house, so that Uncle G's who are above 52 years, they can drop a urine on that particular sample, and they can come up with this, kit by this particular thing. So we wanted to bridge the gap here. We need to peg it. Unfortunately, coming from a private institute, we are not that particular fortunate, to get those particular samples. But I see three grand challenges here. One is the partnerships. We need to bridge the gap between clinicians and scientists. Engagement. We are limited by access, the data, which is very important. And not necessarily we need to employ the phenotype first approach, but it should be the genotype first approach, which means there need not be mutations that might be specific to that particular phenotype, specific to prostate cancer, that would also be largely influencing other entities as well. So I'll probably skip this, and I'm grateful to 4Gs, Galaxy, my gurus, Gandhiji, and of course including Gyan, he's also G, and Genomes. And I'm the founder of BioClose, which is India's largest bioinformatics society. Last year we had Sir Richie Roberts. This year we are going to have Emmanuel Charpentier, where we are going to organize at Institute of Life Sciences in Bhubaneswar sometime in November. The dates will be announced. And thanks all to my wonderful collaborators and clinicians, and my Ph.D. fellows. 5 of them are graduating in prostate cancer and rare disease research, that we are working on. Thank you, Gyan and Chandana and Rachel and everyone.
So thank you very much. I think we should have a very interesting futuristic talk. There would be at least space for one question that you would please.
Thanks a lot for a nice talk. So I'm also glad to see different topics which are covered in this session, especially the single cell data, because this is specifically my lab is interested in Germany. We work with multi-omics and single cell data, including single cell data with brain disorders. So this is something I will also talk about tomorrow in my presentation, and I am really happy that you set up the kind of base for us. So coming to the question quickly, so I was wondering about this data set or the multi-omics you are generating. Is this longitudinal data you are following up in your part 1, part 2, and part 3? Because that would be really interesting.
Unfortunately, longitudinal is a big, I would say, it's a big headache in India, because we have about 3 clinics. Getting these particular samples and the follow-up cases has been a kind of a great concern. But at least out of 106 so far from the samples that we have in the registries, at least 26 of them have been the follow-up cases, and we have been trying to maintain the longitudinal cases. But we're just trying to build upon this. And anyone who is welcome to help us can join our consortium. You're more than welcome.
Nice. Thank you. So thank you very much for such a very nice, interesting talk.
Thank you, sir. I would request Professor Rajiv Raman to come over and present Dr. Prashanth with a memento. I would request the session chair to have a brief summary of the session, please.
So I think we are moving in very right direction. We have this session telling us about the potential of technology in disease management and this diagnosing of diseases, and that's what exactly perhaps all of us would be looking up to. And when you look at